I will be talking about Adalurin, which is a new treatment for DMD. It has a lot of advantages over the other treatments, and I'll be highlighting those as I go through. It has some drawbacks, and I'll try to point those out as well. One of the main advantages is that it's oral, so it's easy to take. It's just a small molecule treatment, so it's pretty simple. The way it works is it'll interact with the ribosome, allowing it to read through premature nonsense stop codons. So what that means, here's a figure demonstrating how it works. You can see on the top normal translation in a healthy individual, and the ribosome will translate all the way through until the stop codon, and you'll get a fully functional protein. In patients with nonsense mutations, you'll get a premature stop signal where the ribosome will stop translating before the normal spot, and you'll end up with an incomplete and dysfunctional protein. With Adalurin, it will bind to the ribosome and allow it to read through that faulty stop signal, and you'll get a fully functional and fully translated protein. So I'll be talking about one of the major studies with Adalurin. It's called Adalurin treatment of patients with nonsense mutation dystrophinopathy. It's the most current study, the largest study that there is right now with Adalurin. So just a little bit of background. About 13% of the cases of dystrophinopathy, so MD, only about 13% are caused by nonsense mutations, and so this treatment would only work in about 13% of the patients with the MD. So that's one thing to keep in mind. It's very promising, but that's still a large majority of patients that will not be able to use this treatment effectively. In general, there's a lack of large-scale, randomized, double-blind controlled trials in DMD. Of course, that's the kind of study you want, but it's just hard to get those with certain disease states like MD. So Adalurin has already worked its way through clinical trials up through phase 2A. The study I'm going to be talking about today is a phase 2B trial. So Adalurin has been shown safe and effective in phase 1 through phase 2A. So it's been studied in animals, and so in mice, and in humans, and so far it's looking pretty good. So the study that I'm going to be talking about today is one of those randomized double-blind placebo-controlled studies that there's a lack of, and so this is providing good, solid evidence. It's a phase 2B trial, so it's in humans, fairly large scale. And the study was at 37 sites in 11 countries, so it's a pretty big population, pretty big study. Of note, one of those 37 sites is actually Children's Hospital here on campus. So some of our faculty here at the School of Pharmacy are involved in the study. So one thing that's important to talk about is the 6-minute walk distance, because that's the primary endpoint of the study. So basically a major goal in MD is to keep patients walking as long as they can. And the 6-minute walk distance is a useful test to evaluate the efficacy of certain treatments. Basically, you just have the patient walk as far as they can in 6 minutes, record that, and the further they can walk, the less progressed their disease is. There have been studies to establish that the 6-minute walk distance is clinically meaningful and that actually measures the disease progression like we want to be measuring. So the way the study worked, they stratified patients prospectively by age, use of glucocorticoids, and baseline 6-minute walk distance. So all of the study participants were between the ages of 5 and 20. So it's not a huge range, but again, these patients don't tend to live very long, unfortunately. Use of glucocorticoids, so about 70% of the patients in the study were using glucocorticoids. So that, and the use of glucocorticoids was pretty even across the groups, so that probably did not play into the results of the study. And then the baseline 6-minute walk distance is just stratifying based on how bad the patient was at baseline. Basically, they had a less progressed group and a more progressed group as far as how bad the disease severity was at baseline. So they randomized them 1:1 to receive a low-dose Adalurin, high-dose Adalurin, or placebo, and TID, because Adalurin is a TID drug. And like I said, the primary outcome was the 6-minute walk distance. And they looked at a whole bunch of secondary outcomes, everything from muscle biopsies to just other functional measures. So the results of the study. So basically, all the groups look the same at baseline, which is good, of course. Like I said, about 70% of patients in each group use the glucocorticoids. 92% of those 72%, or of those 70% use glucocorticoids daily. So there are a lot of people who use glucocorticoids, and if this drug does get approved, it would not be an issue using glucocorticoids for symptomatic control and using your Adalimumab to treat the disease progression itself. And there are 3 types of nonsense premature stop codons, and all 3 were represented in the study. So all patients with nonsense stop codons would be, the results of the study would apply to all patients with the nonsense stop codons. So safety and tolerability, good news, the drug compliance was greater than 97%. So the Adalimumab was extremely well tolerated, which is good news. And the second bullet point is just basically saying that they were measuring Adalimumab concentrations in the blood after taking the dose, and they were able to correlate those concentrations with the drug that was taken. So in the future, if we need to monitor the level of Adalimumab in the blood for any reason, we should be able to do that in a fairly easy, consistent way. So as far as efficacy goes, basically the primary and secondary endpoints all favored the low dose of Adalimumab. Interestingly, the high dose was not any better than placebo, which seems a little weird that the low dose would work and the high dose would not work at all, but that's actually consistent with what's been seen in animal studies and previous trials. And the separation between the Adalimumab and the placebo occurred early and stayed consistent. So Adalimumab worked quickly and stayed effective throughout the study. Here's a chart from the study. You can see on the x-axis you have time, on the y-axis you have the change in that 6-minute walk distance. Remember, the lower it goes, the more advanced the disease is and the worse off the patient is. So you can see on the bottom, the high dose of Adalimumab and placebo, both dropping pretty quickly. At the end, they pretty much end up at the same spot. So the high dose is not really any different than placebo. And then on the top, you can see the low dose of Adalimumab, and you can see that's pretty different from the bottom 2, and that the low dose of Adalimumab was really helpful in preventing the decrease in functional capacity as far as walking goes. So conclusions from this study. So Adalimumab low dose appears to show a clinically meaningful benefit, and consistent with the activity of Adalimumab, treating that nonsense stop codon and helping the body produce functional dystrophin, the Adalimumab treated patients were less likely to lose walking ability. So the low-dose Adalimumab substantially slowed the disease progression. Again, back to that chart, the low-dose of Adalimumab, they progressed a lot slower in that loss of walking ability. And interestingly, the Adalimumab was more effective in the more advanced disease. So at the beginning I said that they stratified patients based on their baseline 6-minute walk distance, and actually the patients with the lower 6-minute walk distance, so the patients with more advanced disease at baseline, the Adalimumab was more effective in them than it was in the patients who were better off and less advanced disease at baseline. So where are we currently at with Adalimumab? So it has conditional marketing authorization in the EU under the name Translarna. As you can see, there's a box of it there. So it has orphan drug status in the US, granted by the FDA, so that just basically means that it treats a disease that is undertreated, that only affects a smaller number of people, and so there might not be financial incentives otherwise to produce a drug to treat it, and the orphan drug status basically just provides incentives for companies to develop drugs to treat those rare diseases, so tax breaks and expedited approval and things like that. Right now, you can only get Adalimumab through clinical trials in the US because it's not approved yet. They haven't submitted an approval for application yet because, like I said, the study I just presented is the most recent, and that's 2B. So, they still have to do phase 3 trials and all that. There are some websites advertising Adalimumab or Translarna or PTC-124, which is just another name for it, but none of those are legitimate because the drug is not approved here in the US. So in summary, for the whole presentation, dystrophin is needed for muscle cell membrane stability. Duchenne muscular dystrophy is caused by a faulty dystrophin gene, leading to the production of a non-functional dystrophin protein. And the currently approved therapies for DMD don't treat the underlying cause, they just help with the symptoms. There are efforts underway to find therapies that will treat the underlying cause. SMT-C1100, it's a small molecule that compensates for loss of dystrophin by increasing levels of utrophin. Another option is AV4658, which induces expression of dystrophin within the treated muscles. And then finally, the one I just talked about, Adelerin, is a promising new therapeutic option that actually treats the underlying cause and allows your body to produce more dystrophin. That brings me to our research proposal. So earlier we talked about how the gene therapy with the mini-dystrophin was not working because basically the proteasome was chewing up the mini-dystrophin that was produced. So we want to look at if you can give a proteasome inhibitor with the gene therapy delivered in mini-dystrophin to see if that will be an effective treatment for DMD. Because we already know that gene therapy with the mini-dystrophin will result in mini-dystrophin production in the muscle, but it's just broken down. So we want to see if we can inhibit that breakdown process and have an effective treatment for DMD. Those are references. That's it. What questions do you guys have for us today?